Thank you very much for this lovely presentation, and thank you all for being here for our presentation, which is entitled Mandibular Morphology in Neolithic and Bronze Age Austria. So, farming and animal husbandry were introduced in Central Europe around 5500 to 5400 BCE by populations from the Near East and Anatolia. Ancient DNA studies have shown that those incoming populations had mixed with the local hunter-gatherers, thus resulting in the emergence of the Central European linear band ceramic farming communities. In addition to changes in population structure, isotopic studies have shown some contrasts between the preceding hunter-gatherer diets and the LBK diets. So, for the Mesolithic diet, it was mainly composed of meat protein from wild terrestrial species like the red deer or the wild boar, by some wild plants like seeds, nuts, and tubers, and freshwater resources also played an important role in the Mesolithic diet. As for the Neolithic diet, it was mainly composed by meat protein from domesticated herbivores like cattle, and by domesticated plants like spelt, wheat, or barley. So, ancient DNA studies have also shown that in Central European, late Neolithic, and early Bronze Age, there was a mixture between the local pre-existing Neolithic groups and incoming populations from the Eurasian steppe. This contributed to the emergence of the Central European corded-ware communities around 2800 to 2200 BCE. So, isotopic studies have shown some differences between the Neolithic diet and the Bronze Age diet, being the most significant the higher intake of animal protein, possibly due to greater consumption of dairy goods. So, as we have seen, transition to food production led to changes in the food preparation techniques, which in turn led to dietary changes. As a result, food stiffness decreased significantly, reducing in turn the muscle strength necessary in mastigation, and possibly leading to changes in mandibular morphology. At the same time, population dynamics like migrations and population history can also impact populations' biological features, and prior studies have already showed that have showed that significant changes in population structure can have a high impact in mandibular morphology. So, knowing this, and since early Middle Neolithic and late Neolithic, early Bronze Age Central European populations have different structures and diets, and knowing that mandibular morphology can be impacted by both factors, our research question is, are there morphological differences between Neolithic and Bronze Age Austrian mandibles? So, some differences are expected, and to test our hypothesis, 42 mandibles, 27 from the Neolithic and 15 from the Bronze Age, from 5 archaeological sites in Lower Austria, as you can see in the map, were analyzed using geometric morphometrics. So, as for the methods, first, a large sample of more than 100 mandibles from the collection of the National History Museum of Vienna were digitized using a surface scanner. Then, all the incomplete specimens were virtually reconstructed. And finally, 21 landmarks were placed in the 3D images from the mandibles that were sufficiently preserved for the morphological analysis. Finally, we used the R package geomorph to estimate the location of the missing anatomical landmarks when necessary, and to undertake standard geometrics, morphometrics, and statistical analysis. So as for the results, no size differences between the Neolithic and Bronze Age samples were observed, as you can see by the graphic. And concerning morphology, we performed a PERMANOVA using all the PC scores, but the test did not detect differences between the two groups, as you can see by the p-value. In accordance, we can see in the graph, as we can see in the graphic, the principal component analysis relative to PC1 and PC2 scores do not show morphological dissimilarities between the two samples. Interestingly, the results suggest larger morphological variance in the Neolithic sample than in the Bronze Age sample. Finally, we did a morphological disparity analysis using the Procrustes distances variances, and the analysis did not detect statistically significant differences between the two groups, as you can see by the p-value. Nevertheless, the morphological variance is greater in the Neolithic sample, as we can see here in the table presented. So, as for our final remarks, contrary to what was expected, no morphological differences were found between the Neolithic and Bronze Age samples. Even though our results do not find morphological differences between the two populations, sorry, I passed one. Future studies with an expanded sample, including increased chronological resolution and other Central European individuals, will provide further insights about the impact of this transition on present-day Austrian mandibular morphology. And now, yes, finally, interestingly, larger morphological variance was found in the Neolithic sample, despite not statistically significant. It is tentatively suggested that this may relate to population dynamics like the founder or bottleneck effect. So thank you very much for your presence, and I would like to thank also to the organization committee of the conference for accepting our presentation. Thank you very much.